Welcome to the Bioprocessing Unfiltered podcast. Each month, we host conversations with the researchers and leaders tackling and solving the day-to-day challenges of the bioprocessing industry.
So I'm Alois Jungbauer, and it's a pleasure for me to do the podcast Bioprocessing Unfiltered with a very renowned person in the field, Professor Georges Belfort. He has been around for several decades, an outstanding expert in the field of bioprocess engineering. But I leave to him to actually talk about his career, and then later on we will actually elaborate on the latest problems in bioprocess engineering in respect to advanced therapeutics and biopharmaceuticals. George, please.
Okay.
Yeah.
Thank you very much for inviting me and for having me join you. It's my pleasure to be here. I'm a professor at Rensselaer Polytechnic Institute for 47 years. Rensselaer is celebrating its 200th anniversary last year. It's the oldest English-speaking technical engineering university outside the military in English. I have to be careful. And I've worked on different fields, but mostly my specialty, if I have one, is how to purify materials, whether it's desalination, proteins, and now RNA. Those are the three areas we've been working on and focusing on in my lab.
Yeah, so you mentioned RNA, mRNA. Yeah? I think this became very popular the past 5 years upon the pandemic, where they have successfully produced mRNA-based vaccine. Yeah. And I think since then you have realized that there are shortcomings or deficiencies in the bioprocessing and purification of mRNA. Yeah? Could you explain us a little bit how you came across why people use, let's say, concepts which are not fully adequate for mRNA purification, and why mRNA purification is different to proteins or, let's say, other biomolecules?
Yes, I can try. So, during the past 20 or 30 years, people have focused on the single most important pharmaceutical drug produced by biotechnology, which is monoclonal antibodies, which were discovered at Cambridge University in England and developed immediately in the United States. And the question there was, how can you build a process that everybody could use? And the name for that is a platform.
Yeah.
And so platforms came up, and generally when you purify proteins, and I'll come back to the RNA in a minute, I just want to lay the groundwork because all of us came from the protein field, and then suddenly we started seeing this change and how RNA has saved some over 20 million lives, probably including mine and yours, and that we take these vaccines and they're extremely effective. So the protein development over these many years was very simple. They decided to grow, you can make proteins either in cells, and more recently we've been looking at making proteins without cells in cell-free media together with a group headed by Jim Schwartz at Stanford. But we would do the purification, and other people do the upstream. That is, upstream meaning the synthesis of the material. And so hybridomas were developed, originally discovered in England. These are spleen cells that have been combined with cancer cells. The cancer cells have infinite life, spleen cells don't, and they make these antibodies. And so this was a major breakthrough in the I forget exactly when was it.
No, 1980s, 80s,
yeah.
early 80s, yeah.
And then after that, they started wanting to manufacture it in large scale. I think over about 70% of all major drugs today are involved with monoclonal antibodies. Anyway, the idea in producing them, so they are generally produced in our body at quite high levels. And what happens is we wanted to emulate that. So we built a hollow fiber reactor in which we put the cells on the outside, these hybridomas, and then we added the food or the nutrients in the feed along the ball, and we were able to get very high levels of antibody produced by these cells. I remember distinctly people coming from MIT and from Cornell to visit us to see how we did that. And then I think people decided that was too slow, or the scale-up was limited, and so they started making them in bioreactors, big steel reactors. And I don't want to go through the whole story, but what you do is you take those cells, you first spin them down, remove them either by centrifuge or a membrane, and then you pass it through the most important step, that is protein A. And protein A is a molecule that was discovered about that same time that basically binds to the Fc portion, that is the leg of the antibody. The antibody is a Y, and at the leg of the antibody is called the Fc portion. And the end of the Y, the end part, are 3 loops on each, and those loops are the binding domains. And so anyway, these antibodies are about 155 kilodaltons. So to answer your question, and so when the pandemic, I forgot, so they were captured by protein A, and then there were other post-translational tests they made through ion exchange. They clean it up, and they have this product, both with chromatography and with membranes. Now, chromatography is the prime separation mechanism, and has always been with proteins. Membranes are the cleanup version. They clean up at the end or they clean up at the beginning, but they aren't the prime step. So now comes the pandemic, and suddenly the 2 companies, Moderna and BioNTech in Germany, started. They got the sequence from China in January. I think it was January 10th. January 10th. And then it was 41 days that Moderna sent the first vial to NIH to be checked of a vaccine material that they felt could be used as a future vaccine. It was basically purified RNA. In that very short time, the question was, how do you purify this material? How do you first synthesize it and how you purify it? So I told you that monoclonal antibody, which is my control, was made out of cells made them, hybridoma cells. But the easiest way to make RNA, messenger RNA, these are linear single strands of RNA, like a piece of string, and it has segments in it that are folded into bulbs and whatever, and it has some double-stranded component, but mostly RNA has lots of hydrogen bonding components from the building blocks, the 4 base pairs. Proteins are built out of 20 building blocks, so they're more complicated. RNA is built from 4 building blocks and less complicated. However, this molecule is gigantic. It's 9 to 10 times bigger than a monoclonal antibody. And that's not trivial, because if it's very big, it doesn't have a diffusion coefficient. That means it can't diffuse well into beads. Remember I said beads are the dominant separating media. So what they did was they drilled holes through beads. So they are porous beads, or they made beads with bigger pores to try to get these materials in. But when you do that, you lose surface area, and there are other problems. When this material, these very large RNAs, get into a bead, what happens is they could basically occlude or inhibit more coming in, because they could be at the entrance, and there are lots of problems.
This material would really block.
That's
They
correct.
would block the pore because
That's correct.
it's big enough, because they would block.
They could block it, so you don't use all this area to bind, like you can with proteins, which is much easier. They're much smaller and they can get in. So the question was, what did they do? Well, they had no choice. They had to do it really quickly. And so they looked in the lab and they saw, there are two methods that we could use. We could put, so I have to go back and tell you what RNA looks like. Messenger RNA has several components to it. Remember, the reason we need it is to clone a protein inside your body. So when you put the messenger RNA inside, it gets read, and it can read off the protein that you want to buy. And so what generally we're doing with this is to clone the spike protein on the outside of the COVID-19 virus. That spike protein you want to teach your body to build up resistance to, so that when the real virus comes, you know all about it. You can fight it. So I think the idea was we better use high-performance liquid chromatography, high pressure. It's very high pressure. It works in the lab really well, but it's really complicated and it's really expensive to make very large-scale systems. So the real breakthrough was that the U.S. government gave over hundreds of millions of dollars, maybe billions of dollars, to these two companies, so that they didn't care which technique to use. They used only the ones that worked. They used scaled up ones, even though they were expensive, and they used that one. But they used the other one, the oligo DT, also.
But it was actually there was a rush, yeah. If they would have time for a year or two,
then they could have done slowly, yes.
If they could have done slowly, they had to take what was available, yeah.
Yeah, I
just to not to blame them.
No, but I think they had to do that to get it quick, to save lives. Because the people were dying all over the world.
Yeah.
So you're right. They had to do this. They took whatever they could.
Yep.
So the two things they took was they took an affinity bead with a pause in it, and they put oligo DT in it. They grafted it on, and T binds to the tail of the messenger RNA. Messenger RNA is like a dog. It has a tail, and the tail is made from one of those base pairs, A. And it's like fishing, when you want to throw it in and you catch the fish you want. And it's that tail that is caught selectively by the oligo DT on these beads. And so it worked beautifully, except it's only about 1 to 5% efficient. 1 to 5% efficient.
Yeah.
So that's very expensive. As one person once said to me, we're spending millions, if not billions, on these beads.
Yeah.
And so that's the story of where I come in. So when you ask me, how does it work? So we decided, I've always tried to move orthogonally to where everybody else is going. If everybody's working on this problem, I said, that's fine. There's too many people. I want to go in this direction. So I tried to go in 90 degrees from them. And I thought, I'll write a short grant. Actually, I didn't even know about it. My colleague, Todd Probitzian, walked in my office and said, George, the U.S. government is going to give money for working on the purification of RNA. Why don't you write a grant? So I said, all right, what did I write on? So I said, I write it on using membranes instead of beads And it turns out it was a very good move.
Yeah, but it's clear because you had many years of experience on membranes, and you know
with proteins we use them
with
too,
proteins, but you know how membranes work, yeah.
and
so
what's wrong with them, and what their limits are, and the challenge,
and how to chemically modify them, and so on and so forth. Yeah, so I think
so that's what we did for 20 years. We were modifying membranes. So we thought we could easily modify them and put the oligo T in a membrane and push this very large molecule through this porous media and not allow it to diffuse. So you just push it through, capture it, and see if it works. So there was an interesting problem. I wrote the grant. It was 4 pages. I'll never forget it. And I think I forgot about it. And about 3 months later, I got an email to say, you've got the money. And I couldn't believe it. There were several problems. I never had messenger RNA. I never had the right membranes. I never had the oligo DT. And I never had a team, and we had no RNA lab. So we had to solve all those problems. I didn't sleep for a few nights because I wanted the money, but I didn't know how to get all of this. So I immediately started phoning CEOs of companies, the top person, not the second and the people I know. I didn't even know some of these people. And they all said, we'll help you. Everyone. It was unbelievable. And IDT was the best. They said, we'll give you all the oligos you need. We'll give them you with FAM on them. FAM is a fluorescent tag, so that then you can see the tag with what you're binding and how you bind. So we worked with them for many years now. We have an agreement that just ended about a month ago where they gave us everything free for 4 years. Now they want us to pay. But anyway, it's been wonderful, and they have been wonderful. So I think I then phoned up the membrane company, Cytiva, and they said they have a group in London, and they'll give us what are they? Whatman membranes.
They
are cellulose membranes, and they are excellent. Then I phoned up and I got in touch. I was a consultant some years ago with a company in Japan, and they make hollow fiber modules for virus separation.
So this is Asahi.
I know them very well.
Yeah.
Because I was a consultant to them in America. And so they sent me membranes too. So suddenly we had the membranes, we had the oligos, and now we had but it's not trivial to build an RNA lab. And so the question is, what am I going to do?
But what is the challenge what is the challenge for the RNA lab? Yeah, what is different?
RNA does not live long in the environment. On our fingers and everywhere we are Pac-Man that eat RNA. So we have them they're all over. So you can't just start like proteins, put it in a glass and start running. You have to wash it with special water, get rid of the RNAs. You have to do everything on the table. You have to it's a special lab. However, I was very lucky. My wife is a famous RNA person. So she said to me, I will arrange for you. Come across, bring your team. And we went across 16 miles to the other university where she works, at UAlbany. It's just across the road in Albany, and we're in Troy. So it was easy. We drove across, and there's an RNA institute there. And the director, Andy Berglund, is a wonderful person. And he said, George, we'll help you do everything. We'll tell you what to buy on your grant. We'll teach you how to run a lab. You bring your students here. We'll do it all. So they did it. And within 2 months, we were running with a complete RNA lab.
So your network, your connections actually accelerated the project. Yeah?
I don't know. I was lucky. They were nice people.
No, I think
there were lots of very nice people. Yeah. They could have said, I'm sorry, find somebody else. Actually, one company did. When I asked them for RNA, they said, you have to ask somebody else. So we did.
Okay.
So,
Yeah.
but we got everything, yeah. But it was just lucky too, I think a young person would hesitate to phone a CEO that they don't know in a company. I mean, what happened with IDT was they're in Iowa. They're the single major producers of oligopeptides in the United States. So I asked my wife, who's the top person? She said, I don't know anybody, but they're the people you want to go to. So I phoned up the CEO. I said, I don't know you, but I've looked up your background. You're a biochemist. Now you're in the business world. I need your help. And he said, I'll help you.
Great. This is
So
great.
that was it.
Yeah.
So it was wonderful. They were wonderful.
No, so I think what we have seen also with your presentations, your students' posters here at the Bioprocessing Summit, yeah, you have really progressed a lot, yeah. You are not yet fully there, yeah, but it's really impressive what you have achieved in the past couple of years, yeah. Could you tell us what challenges, what open scientific questions you would now tackle in this, let's say, next period? Yeah, because you successfully developed a membrane with oligo-TT for affinity purification. We have seen you have peptides selective for messenger RNA and so on and so forth. So what is left?
There's a lot left. And by the way, none of it was done by me. It was done by my students and postdoc and my collaborator. So I have to give them credit. So my collaborator, without him, we couldn't do the peptides. His name is Pankaj Karande, and he's ex-student who now is a postdoc in our group with him. He's also in our group. And her name is Ming Yang or Claudia Hu. And so she did all this discovery. And what we did was we took her peptides, grafted them on, and the student that did that is Riddy Bunnick. And then once we've got it on the membrane, we now had to test it, and that is Thomas Newman. However, that wasn't enough. We also wanted to know, to prove that if we produced or discovered a peptide or a ligand, like I call a hook, if this hook can catch the right thing, we need to find out. So we in my group have done for 20 years. Actually, 1989, we published our first paper on single molecule force measurements in PNAS. So that was to look at lysozyme, see how it actually sits on a surface. Now we can do it very easily in the lab. The student's name is Surya Kala, and he actually can measure forces between a RNA with a cap or without a cap and a small peptide or an oligo-DT, anyone, and we can measure the forces. So that was one of the posters we showed. Many people are very interested in it because it's a direct measurement. And you can also change if you want to know not only how the forces are, but how you can get it off, elute it. Because we need to elute it to catch the product. You can change the buffers and find the optimal buffer.
My question is now, what is needed to translate your findings, your research into a product or into, let's say, into the bioprocessing industry?
Well, I think there's several things that could be done. First of all, I think the big product would be, we sort of think, I was approached today about starting a company by a British scientist who said to me, he started a company, why don't you? So I said, well, I'm going to speak to Bob Langer about that and see if we could start a company. But really, I'm not so interested in money. I'm interested in technology and discovery and how to do research. So I think the first thing is we need to make these membranes commercially available. If they're not commercially available, it's not good enough to have it in a university. And I don't know if the companies are interested. If they are or they're not, we will see. So we also have a method, a new method, with Thomas and Rudy, in which we don't use a hook. It's got no hook, but we can still catch the fish. And we do it by actually using the difference in the properties between the unwanted immunogenic, that means causing an immune effect in the body, double-stranded versus a single-stranded, which you want. That's the vaccine or therapeutics. So I think the future is we need to have a membrane. Now, the second thing is we need a good module. The modules need to be designed. Module is a box in which you put the membranes in.
Yep.
It turns out these modules that are built today have quite a lot of dispersion in them. And dispersion is a fluid mechanics mass transfer problem in that you get mixing. And you don't want to get mixing because for affinity, you want it to go through evenly.
You want to have the black flow.
That's right.
Yeah.
It has to go through the pores. Even you can go mix, have some going through, some not going through.
Yeah.
So we have designed and patented a new small test cell, both for hollow fiber. It's the first hollow fiber replacement. You can put a second hollow fiber and you don't throw it away.
Yeah.
Currently, you have to throw them away because there's the companies like the Japanese company, Asahi, they sell you a hollow fiber, but then you have to throw it away. We can take the hollow fiber and replace it. So we have a patent on that, and we built it, and it works.
But I'm interested. Companies have approached you already about your findings, discoveries, and so on, or not?
Yes. Some have. I can't tell you who, but I don't want me to say anything.
No, it's fine. That's
but
clear,
some have asked us, what are you doing? Can you work with us? And so that there are other possibilities. By the way, I've told you oligo T is a hook. Peptides can be hooked. And our peptides, we can make a hook against any fish we want, any molecule. And thirdly, the non-ligand method. But there is a fourth way that people are talking about, and that is to use organic chemistry to make small organic hooks. It may be cheaper. It has a much bigger monomer library, 500 instead of 20 peptides or 4. So it's possible. And there's a group in England that approached us. There are other interesting things.
But I think your peptides already are very promising.
Yes.
That's what I see. This is a really promising affinity ligand.
Yes, that's what it is. But
there
is competition. There is some competition.
Competition from other groups or other companies?
Well, there's a company in San Francisco that does that. But I don't think they do what we do. We don't only look for the peptide. We do a full analysis of the simulations of the molecules binding. So we want to look for binding. And then we start to see that half the peptide is not binding. So then we change the amino acids to make it bind correctly.
So we do have the binding efficiency to improve.
That's correct. That's it.
Because this would reduce the costs of that.
And also reduce how much you need.
Yeah, it's clear. Yeah.
But there is one endemic problem with these peptides. So, and also oligo-DT and all of these binding systems. And I just want to tell you what it is. When you have a very large molecule with a tail, like a very large fish, let's say it's a whale. You're catching a whale. It's got a tail, and you can catch it by the tail. So and you want to catch it on a surface, say on the carpet. What you do is you cover the whole carpet with these ligands, and they will catch the tail. But only 2 or 3 of them will catch the tail. The other 99% is in the shadow underneath.
Yeah.
It doesn't do anything. You've wasted your time, your money. But the question is, and this is what we're looking at now, can you minimize the amount of binding, and can you make less of it, and then you can actually bind only and not pay for so much and still have statistical binding. So we're looking at that very carefully now. How can you optimize and minimize the cost? But I do want to tell you there are two other opportunities in this whole field that's coming up. One is circular RNA.
Yeah.
So there is a new company called RNA Vate in Cambridge University, next to Cambridge University, started by the Nobel Prize winner Venki Ramakrishnan, who actually is a colleague of my wife. So we've been in touch with him to talk about how to purify circular RNA. There are a lot of opportunities there, and it's not easy. Second, when you make circular, sometimes it gets nicked.
Yeah.
And the nicked molecule is identical to the circular. How do you separate that?
That's really that's.
So I think there are ways to do it. I don't want to give away all the ideas, but there is a way to do it using a physical separation. And thirdly, and by the way, you could also bind stuff to the two ends, because the one has ends and the other doesn't. And if you react something to it, you may be able to separate it that way. But we're looking at this problem. And then there's self-amplifying RNA.
Yes.
I have a big worry about that molecule. I'll tell you why. It's a molecule that you put in, and it has a segment. It's much bigger than the RNA we're talking about. Instead of being 4,000, it's 10,000 base pairs. So it's another 10 times larger. It's massive, 5 times larger. The problem with this is not so much its idea, but the concept is you really want to put a molecule in your body that never stops. That keeps self-amplifying and keeps going. Maybe FDA won't be so pleased with us to go. So it's been approved in India, but not yet in the United States.
George, I think my impression after this discussion with you, what you have shown us, you have really developed enabling technology to reduce the cost of goods and manufacturing costs of mRNA molecules, which are have a great future and also may really benefit mankind. Not only as a COVID or influenza vaccine, cancer vaccine, and so forth. So I thank you for actually
Thank you very much for inviting me, and thank you for us. I hope I gave you some ideas.
I think these were brilliant ideas about mRNA purification and downstream processing.